Thank you everyone for joining us today. This is our bi-weekly Wednesday seminar in the Open Source Neuro series, which is part of the Worldwide Science Initiative. Our series is all about open tools and methods in neuroscience that we collect on our website over at open-neuroscience.com. Don't forget to upload your projects to the website and sign up to give a talk of your own. We welcome your contributions and love if you can follow us and spread the word. Today we have the pleasure of hosting Dr. Daniel Aharoni. Dr. Aharoni is currently an assistant professor in the Department of Neurology at the University of California, Los Angeles. He received his PhD in physics from UCLA, working in particle physics, before shifting focus to neurophysics, and continuing on in UCLA as a postdoctoral fellow under Drs. Kach, Silva, and Golshani, where he spearheaded the technical development of the open source UCLA Miniscope project. Dr. Aharoni's lab lies at the intersection of engineering, neuroscience, and physics. Specifically, his lab focuses on applying tool development methodologies from engineering and physics to address current challenges in neuroscience and medicine, with three main goals: to contribute to understanding circuit-level neurological function, to bridge the gap between specialized tool design and the intricacies of modern neuroscience, and to promote equitable access to transformative tools and techniques in neuroscience. Dr. Aharoni recently won the NIH Director's New Innovator Award to develop long-term neurobehavioral recording and real-time processing platforms for naturally behaving animals. Please feel free to ask any questions in the YouTube live chat or join us in the Zoom room for a discussion with the speaker. Thank you for being with us today, Daniel, and please go ahead.
Great, thank you so much for the invite and the nice introduction. Definitely feel free to interrupt during the talk, anyone, if you have questions, and then we can chat also afterwards. So I'm here to talk about the project that I've been working on since a postdoc, and now my lab leads kind of the technical development of this project, which started now like maybe almost 8 years ago, which is the open source UCLA Miniscope project. And our goal is to really advance the techniques that allow us to record and manipulate neural and behavioral data in animals that are as naturally behaving as possible. So this usually sort of gets implemented as some sort of wearable miniature microscope that can be mounted on the head of animals down to the size of a mouse. And then those mice or larger animals are able to freely behave and perform behavioral tasks while we are imaging neural activity with single cell resolution, along with recording behavioral measures to process later on. So the general, I think, structure of my talk is I'll give an overview of what our UCLA Miniscope project is. I'll talk about sort of like the current state of the tools we're developing, and then some of the work we're doing now to really push some of these techniques forward, and then where we probably imagine miniature microscopes and this type of work to go in the future, and then spread throughout. I'm sure I'll mention what I think is the really key importance of open source development of tools and techniques in neuroscience, which I think it can't always be done, and there's absolutely a place for commercialized devices for all the things that neuroscientists need, but there's also some critical importance for people that work on open source tools. Okay, so this would be good. All right, so the Miniscope project, we can think of being the hardware and system itself, and that consists of usually a small head-mounted epifluorescent miniature microscope. So this is a single photon excitation microscope, very similar to what you would see as like a large tabletop fluorescent microscope. It just has everything that's not necessary removed, and then the remaining components made as small as possible. That is on the order of 3 grams or less to be worn comfortably on the head of a mouse. And then the head-mounted microscope needs to be able to get powered and get configured and send high bandwidth imaging data to an off-board custom data acquisition board. And we do that through a single flexible coaxial cable. So coax cable is just a single inner conductor with some insulation and then a grounded shield around it. So our system's on the general cabling that comes off the animal's head is a 1 millimeter diameter, super flexible commercial coax cable. But you can run our systems down to like a 0.3 millimeter diameter coax cable. So it's effectively kind of like dental floss coming off the animal's head. And what we've seen is that as long as you're within a reasonable weight, animals can perform very naturally with a little bit more weight on their head. But the thing that affects their behavior the most is the flexibility of the cabling that comes off the animal. So if you imagine you have some big heavy cable that provides a lot of torque to the animal's head, it can really disturb their natural behavior. So we've done a lot of work in really minimizing that impact to now the super thin flexible coax cable, which I think is critical for a lot of experiments. So the system connects to the data acquisition hardware, and then that's really the Miniscope system, miniature microscope and some acquisition hardware. And then that system usually connects to a computer running our open source software just over USB. And then the system, of course, you're not going to generally just be doing neural imaging. You're going to want to record some sorts of behaviors while doing neural imaging. So the system then can be synchronized with behavioral cameras and other behavioral types. So sort of just as important to us as the tools we develop is to develop resources that anyone in the world can access so that they can build and understand and use these microscopes and tools we develop so that they don't just sit in our labs or in our collaborating labs. So we probably spend just as much time on building these resources. And I think about them as kind of being spread over two groups, and then a bunch of people kind of sit somewhere in the middle of these two groups. So most neuroscientists sort of would just be considered like users of these systems. They don't necessarily need to download the source code or modify the circuit boards. They just want to be able to go online and watch some video tutorials and guides, be able to purchase all the individual components of the system, assemble it, and get up and running. So for users, which again are the vast majority of neuroscientists that use our system, we provide all the fabrication files and the compiled source code and the 3D files if you wanted to 3D print the parts or get them machined. But just as importantly, we provide all of our work for people that want to take what we've done and really build on top of it. So for developers, we provide all our work in terms of source codes and circuit schematics. We try to work as much as possible in free and open source design software so that there isn't a technical or economic hurdle to starting to look at our designs and modify them. And being able to provide all the work we do in kind of this base format really, I think, has accelerated other advancements in miniature microscopy. So there's probably a dozen or so other modifications or versions of miniature microscopes that use components that we've initially developed. So it's a good kind of jumping off point for building your own tools. And so we initially started the Miniscope project because there's 3 labs I was working in as a postdoc, and they just wanted to, they had some really good ideas for some 3D behaving experiments where they wanted to record single cell neural activity over hundreds of cells. But very quickly, we realized how impactful and transformative these sorts of tools could be. And that, like, given the state of like high quality, low quantity manufacturing and like large commercial investment in small image sensors with really high sensitivity, it really started making sense that these sorts of tools are not too difficult to build. They're easy to understand, and we can take advantage of all this commercial investment in development of some of the individual components we're using to really build a very robust system. So pretty early on in the project, we decided that we really wanted to make all of the work we're doing open source and really kind of invest our time to making sure that it's not just open source in the title of the project, but that we are really enabling the neuroscience community to be able to use these tools. So in 2016, we opened up access to kind of our first design of a miniature microscope. Since then, something like, I think the number is probably closer to 3,000 miniature microscopes have been built. Again, this is an open source project, so we can't really keep track of all the miniature microscopes that people build, because someone could go to their university machine shop and get the parts machine and go to their favorite PCB fabrication house and get the circuit boards made. But of what we know, we're probably in the ballpark of 3,000 Miniscopes. That's probably over 600 to 800 labs across 18 countries. And this is a map of sort of the activity to our wiki page and universities and cities that are using our lab, our tools. And then additionally, COVID slowed this down a bit, but we try to do in-person workshops and now some virtual workshops to train people how to use these tools. So to understand the theory behind the microscope, understand how to assemble it, do surgeries, animal handling, analysis. Usually this used to be kind of 15 to 20 person 2-day in-person workshops that are very hands-on. We sometimes do some larger workshops. And at the end of November this year, we're doing a large virtual miniature microscope workshop. So if you're interested, you can Google, Miniscope virtual workshop, and a link to register will come up for that. Okay, so what does the system look like? So this is the original miniature microscope we developed that we openly shared, which we call a version 3 Miniscope on a mouse. It was around 3.3 grams. And it allows the mouse to freely behave in this open field while we are imaging neural activity. So the microscope, we'll see in a second, but just shines blue excitation light that illuminates all the tissue underneath the optical implant or cranial window. Cells that have been labeled with some calcium reporter, usually GCAMP, that are active will fluoresce green, and that green fluorescent light travels up through the microscope and forms an image on an image sensor, really like what you would sort of find in a cell phone if you opened it up. So I'm not sure how well this will show up over Zoom, but the top right is just the raw video streaming out of this Miniscope. We're imaging GCAMP6 fast in the CA1 layer of the hippocampus. And so you can look, if you look closely, you can see all these little kind of spots light up, and those are cell bodies. When they're active, they will fluoresce. We generally do some kind of minimal processing to make it a little bit easier to see by eye. So the bottom right has had motion correction applied to it. So because we're imaging in freely behaving animals and the brain has sort of a consistency of jello, the brain wobbles back and forth roughly on the order of like 10 micrometers, so the size of a single cell. And that wobble needs to be corrected before we can do further processing. So using one of many image alignment techniques, we can stabilize the image. And then we did, for this, a very rough kind of delta F over F conversion. So the bottom right just helps you visually see the cells, and the pixels are sort of showing the percentage change of fluorescence. And each one of those dots is a cell body. In this recording in hippocampus, there's probably like 500 or 600 active neurons while the animal explores this open field. And a few kind of, I think, key features of our setup is, well, our newer microscopes are a little bit over 2 grams that give over a millimeter diameter field of view. So you can really, with good expression, image over 1,000 cells in a freely behaving mouse. And the devices we design are really designed for neuroscience scientists to use, and for neuroscientists that we've never talked with to use. So we really try to build things that make doing experiments easier, because like neuroscience is just ridiculously hard to do. Working with awake, freely behaving animals is really difficult to do. We know, for most of their experiments, researchers are going to invest a ton of time in training their animals, doing surgeries to, plant an optical probe or something. And you don't want to get all the way to that point and then have tools that are faulty or fail in the middle, that which can happen, will happen with kind of all tools, but should happen not often. And since our tools are open source and most people build the microscope themselves, if like the mouse chews through the cable, you don't have to be worried about fixing it yourself. You built it once. You can. I think usually people, after they built one miniature microscope, they feel very confident that they can modify it or fix it if something were to happen, compared to maybe some commercial options where if something breaks, you have to ship it back to the company to get it repaired or replaced, and it could, stop your experiment in the middle of it, which we really would like to avoid. Additionally, we have things like the system is commutatable, so you can put it through a commutator and have long recordings where the cable doesn't get tangled up. And then probably very important for many people is our system is just affordable. And this isn't because the system is like not as capable as commercial systems. I would argue our newest miniature microscopes are the most capable open source or commercial miniature microscopes. But what goes into these types of microscopes is just not that expensive. So you really allow me to get up and running with a system with like a data acquisition box and a couple scopes for on the order of $2,000.
Daniel, can I interrupt you for a question?
Yes.
Sorry. Amal asks, what size green lens can be used with this Miniscope?
So you can use any of the standard size grid lenses. So usually what's called a relay lens, relay grid lens is on the order of a 0.5 millimeter to 1 millimeter diameter. They vary in lengths from like 4 millimeters up to like, I don't know, we have some lenses that go up to 30 millimeters in length. And those grid lenses, you basically, which I'll show in a second, you implant them within a few hundred micrometers of the tissue and cells you want to image. And those relay lenses just relay images from inside the brain to right outside the top of the relay lens. And then the Miniscope images that intermediate image. For larger animals, you can image through, kind of the largest standard available grid lens is a 2 millimeter diameter grid lens, which we'll see a little bit later that we use in rats. And for our newer microscopes, which I'll also show, we can image the entire 2 millimeters or even bigger of these larger grid lenses to get really large recordings of neural populations. Yeah, so it's kind of, it should be compatible with basically all the relay grid lenses currently out. Right, so if we wanted to image an animal, neurons on their own don't fluoresce. So you can use transgenic animal lines, but oftentimes you'll do a viral injection of a GCAMP in the brain region you want to image. Those cells get transfected with GCAMP, produce the GCAMP protein, and are able to fluoresce after a few weeks. We then need optical access to those cells. So if you're imaging cortex, which I'll show in a little bit, you can now, with our newer miniature microscopes, just do a cranial window, just like you do for 2-photon imaging, and have the Miniscope mounted right on top of the cranial window and image into cortex. But the brain, it has, you know, its volume, and oftentimes you want to reach deeper brain structures. So in this case, for hippocampus, let's say, hippocampus is a little over 1 millimeter down from the surface of the brain. So in this case, we aspirate cortex above the CA1 layer and implant what's called a relay gradient index of refraction lens, or grid lens. And these lenses are cylindrical lenses with a flat top and bottom, and they relay images basically from inside the brain to outside the brain. And so those give you now access to basically any structure in the brain. And then we will, so the grid lens or cranial window gets cemented to the skull. And then we will cement an aluminum base plate. The lens and the base plate is the only thing that's permanently attached to the animal. And when we want to image, the Miniscope slides into the base plate. You image neural activity while the animal is behaving, and the Miniscope comes off, and you can come back and do this day after day, image the same neural population and track individual cells across weeks and months. So if we put this all together, a recording will look something like this. So this in the top left is a mouse running on back and forth on a 2-meter linear track. We're seeing the red LED on the top of the Miniscope and some simple algorithm to track the animal's position, shown in the white circle. And here we're imaging kind of monochromatic green GCAMP light. But once we go through the pipeline to identify individual cells and attribute fluorescence activity to them, we can pseudo-color and generate kind of this video, which pseudo-colors individual cells, and the brightness is modulated by the amount of fluorescence attributed to the cells. And here, in hippocampus, there's probably 500-600 neurons being recorded here, and some example traces of the fluorescence of 50 of these cells are being plotted on the right. And so this gives you quite high cell counts. Again, depends on the expression and the brain region you're imaging, but in hippocampus, for our older Miniscopes, we average somewhere between 400 and 600 active neurons that could be detected in a 10-minute recording. For our new Miniscopes that have like a 4-time or greater field of view, we can average over 1,000 active neurons recorded, which is really exciting, and kind of opens up the ability to do a lot of additional analysis that maybe you couldn't do when you're limited to like extracellular EPHIS recordings, where you may, in mice, you might get tens of cells in a session. And between sessions, it's difficult, if not impossible, to say the same cell you clustered spikes of is on session 1 is that same cell in session 2. Here, we get hundreds of cells, and because we get the spatial distribution of the cells along with their temporal activity, we can match individual cells across days and now do longitudinal studies and track activity across weeks or so.
So we have
so that's
another
sort of, yes.
question coming in, sorry. Does the green lens implant with the virus injection, or should we wait like 2 weeks after the virus expression?
That's a good question. So people do it differently. The kind of time constraint you have here is like GCamp viral injection will take somewhere between 2 to 4 weeks for the cells to begin to visually fluoresce. So there's a time course there for expression. Additionally, if you implant a grin lens, and even sometimes the case with cranial windows, so when you implant a grin lens, there's an immune response of the brain tissue, which effectively blocks the fluorescence from getting into the bottom of the grid lens, and it takes a few weeks to clear up. So, expression takes 2 to 4 weeks, sort of the inflammation response and the clearing of the field of view takes also, 2 to 3 weeks, let's say. So you would want to time around those things. Oftentimes, people will do a viral injection and lens implant on the same day, especially if you're going to very deep structures where you kind of have to be quite accurate in the coordinates that you implant to. People will head bar a mouse, do the injection with coordinates on a stereotax without moving the animal's head, so you're not moving kind of the origin of the your stereotax. You then go and implant usually slightly above where your injection site is. But you could also do, like for hippocampus, when I think the labs that we work with, oftentimes we'll do a batch of injections just because it's quicker to do 5 injections in 5 mice, one after another, and then come back and do individual grin implants. I guess the only other thing I'll say is that it used to be the case, especially for deeper structures, that some people thought the most optimal approach to give you the highest chance to actually be able to see cells once you're done with doing the injection and implant is to do a GCamp injection, wait until those cells are able to fluoresce. And then when you implant like a long thin grin lens, you image the grin lens as you implant it, and you basically move down until you start to see fluorescence, and that's where you stop, and that's where you cement everything in place. What we have heard from multiple groups is like, while that sounds like a good approach, a lot of groups, their best chance and highest success rate is really to just go in blindly, but with the exact same coordinates. So injection, you don't wait until the cells fluoresce, and you try to hit the exact coordinate you want relative to that injection site with your lens implant. Cement everything. You wait, 3 weeks for the virus to express, for the field of view to clear, and then you image. I hope that answers.
Just one more, thank you for that. Yao asks, what is the typical SBR of those neuron delta F/F, which I guess we were showing earlier.
What is the typical what?
SBR. Oh, I don't know what that means.
I'm not sure what SBR is.
Yao, if you'd like to unmute or tell us in the chat.
Oh yeah, sorry. So it's like you have those like many neurons, delta F divided by the F. So I'm wondering, like, what's the ratio of the delta F and the F?
So for kind of this kind of epifluorescence single photon imaging, while maybe like 5 or 6 years ago, we would use some measure of delta F/F, our baseline fluorescence cannot be detected as the baseline fluorescence of an individual cell like you would do for 2 photon imaging. Our fluorescence is like all this background fluorescence from out of focus cells and neuropil. So we really don't have a good measure of F. So all the processing that's done on this imaging data kind of tries to remove the background and doesn't convert the image to delta F/F. But if you were to convert this, sort of like, what is your signal over just the overall background? It ranges from, I would say under 10% to like over 100%. So it depends on the expression, the quality of the surgery, which miniature microscope you're using to image. But it does put a limit, especially because our background fluorescence or baseline fluorescence is reasonably high. And the sensitivity of these image sensors are lower than what you would have as like a photomultiplier tube in a 2P microscope. You generally cannot resolve individual, like the fluorescent response from an individual spike. So we're usually seeing trains of spikes that generate a large enough fluorescent response for us to see.
Okay, yeah, thank you.
Cool. Okay, so that's a rough overview of miniature microscopes. We're not the only one that even makes open source miniature microscopes. There's quite a few with different capabilities. I think ours is, I'm sure, the most widely used miniature microscope in the world. We really try to support it as well, but many other people do the same. So our newer generation of miniature microscopes, which we call the version 4 miniature microscope, which has been out for a little over a year. These systems, you can go to a couple open source distribution companies that we're not directly involved in, but we work with to make sure that they can source the components that they're building, that they're selling systems that are reliable. And so these are Open Ethos Production Site, OEPS, and lab makers. So there you can, instead of sourcing the individual components, they buy all the things in bulk and put it in a kit for you to assemble, or they even now sell fully assembled and tested miniature microscopes. And we make sure that the pricing is effectively lower than what it would be if a lab tried to source a few of these components, in lower quantities themselves. So on the order of $1,000. But some key components of this newer generation Miniscope is we really took a step back from what we had developed in the past and sort of the standard design of miniature microscopes, and really tried to address some fundamental limitations in that design so that we have a base platform to move forward and design around for all of our new ideas. So some key components is like, this is smaller and lighter than our previous generation miniature microscope, while increasing the field of view substantially, which we'll see in a second. Our older microscopes, you could adjust the focus in the brain, but it was a mechanical slider you had to move to adjust focus. These new systems all now use electrowetting lenses. So a little lens that through software, you can adjust the voltage on the lens and it adjusts the focal plane for you in a very repeatable manner. And then, we use these miniature microscopes, our collaborators do, but we really want anyone to be able to use and feel comfortable using these systems. So we try to make the assembly as easy as possible. Like, these are 3 machined housings, which are easy to get or to make yourself, and you just pop optical components inside them. And then usually what I think for a lot of neuroscientists, they're a little worried about is, one, maybe you've never worked with optics and you're kind of worried about assembling. You can do a surgery in an animal, you can push some lenses in some plastic housings, but oftentimes people are worried about having to solder components on on a circuit board and ruin something. So our newer systems are these rigid flex circuit boards that come, if you got it from an open source distributor, come fully assembled, and there's no soldering involved at all. And when you want to connect a cable, it's just a small, tiny coax connector that you snap on. So they're really easy to assemble. And again, they all rely on these thin, flexible coax cables that make it really compatible with the breathing animals. So as I mentioned, we have this kind of electronic focus. There's just an image of a resolution test slide while we're moving a slider back and forth in software. And we can adjust kind of plus and minus 150 to 200 micrometers. So you can really look up and down your tissue to find the plane that has the best imaging of cells. We also put an absolute head orientation sensor on this miniature microscope and sort of all the ones we're working with now. So this is a 9 degree of freedom gyroscope, accelerometer, magnetometer that merges that data within the chip and just can output kind of an absolute orientation of the chip. And so this is recordings done through our software, but someone's holding a Miniscope, moving it around on the right. And then the left is actually the video coming out of the Miniscope. And that little logo, when it's enabled, rotates with the Miniscope, and then all that data gets saved. So you can do really high accuracy head orientation tracking and analysis while you're doing a Miniscope imaging. Another thing that was really important designing the system is we didn't want to have to rely on using implanted relay grid lenses or other grid lenses. So our old system and the vast majority of other miniature microscopes out there, if you wanted to image cortex, you still would have to do a craniotomy and sit a large grid lens on the surface of cortex, cement that grid lens that now might stick up 5 millimeters, and then mount your Miniscope above that. redesigned the optics so that the kind of standard working distance of the Miniscope is closer to a millimeter. So from the bottom of the Miniscope to the focal plane, the imaging plane, is roughly a millimeter, and this can actually be extended up to 4 millimeters. So this allows you now to just do a regular cranial window surgery that you could put the animal, head bar the animal, put them under a 2P microscope, and then put a base plate on and image with the Miniscope. This now really makes cranial cortical imaging oftentimes easier. A lot of labs are already comfortable with doing these kind of cranial windows. And now, because the working distance is larger, you can reach through the cranial window into, like in mice, usually people image layer 2, 3 in cortex, which is really great. So this is an example recording from a large cranial window by Mega Seagal from Alcino Silva's lab at UCLA. This actually has, so because things are open source, we can swap out different lens components, and this is outlined on our wiki, and you can change the field of view or the working distance or the magnification. So this actually is pushing the field of view of this version 4 Miniscope closer to like 1.5 millimeters diameter, and now imaging across pretty large cranial window in a freely behaving mouse. And so this just really minimally processed, but you can see a bunch of cells light up as the animal is doing some behavioral tasks. But we oftentimes want to go deeper, and so these miniature microscopes are fully compatible with pretty much all relay grid lenses that go from a millimeter a 0.5 millimeter in diameter all the way up to 2 millimeters in diameter. And so this is a recording from Pete Zhao from Payman Golshani's lab. Is actually one of the first version 4 recordings we did a couple years ago, imaging a medial prefrontal cortex through a 1 millimeter diameter grid lens. And so the field of view of the Miniscope is pretty much almost, with standard optics, almost exactly 1 millimeter. So we're imaging the entire surface of the grid lens. And so this is the raw recording, which hopefully you can see some neural activity. And again, if we do some minimal processing to stabilize the recording and do some rough conversion to delta F over F, there's on the order of 1,000 neurons here as the animal is freely behaving. And again, you can go to much deeper structures as well. Oftentimes you would use a 0.5 millimeter grid lens if you go very deep.
So there's another question coming up. Amal asked, is there any option for the dual color imaging with V4 or other Miniscope?
Yeah, we're working on, I don't know if I put it in. At the end, I'll show it if I don't have it in the slide. So our thought behind making this version 4 Miniscope is it really is a base platform for a bunch of things we want to do around it. So one of those is to take this design and turn it into a dual color Miniscope. All the optics in the version 4 are achromatic, so you could, which is an issue if you were using a large objective grid lens. Grid lenses have a ton of chromatic aberration, so we have removed that with achromatic lenses, and it's effectively the exact same version 4 Miniscope, but with a different circuit board that provides dual excitation through the system. And so at the end, I'll show a slide on it if I don't have it here. We basically finished the design, and we're sort of in the final process of validating it in a couple different combinations of fluorophores and freely behaving mice. All right, so another kind of goal of these miniature microscopes we're developing now is to kind of extend the length that you can record for. So before miniature microscopes were developed, if you're doing neural imaging, you would generally be head fixing the animal. And even if you could record for long periods of time, generally you don't want to head fix a mouse or a rat for longer than tens of minutes to get uncomfortable. But now that we can have comfortable wearable miniature microscopes, we don't have this constraint of kind of limiting our recording length just because of the animal's head fix. So our newer Miniscopes are on the order of 15 to 30 times more sensitive than our previous generation Miniscopes. And so this allows us to use much lower excitation light to get the same quality neural activity. And because we're using less excitation light, there's less photo bleaching, and we can extend the length of our recording. So this is a recording very much sped up over 9 hours of an animal just kind of spending 9 hours in this open field. That's just the kind of heat map of its behavior across this open field. And across the 9 hours, we get on the order of like 5% photo bleaching in this recording. And the quality of neural activity right at the, extracted neural activity at the beginning is very comparable to the quality right at the end of 9 hours. We've now improved the sensitivity and optical efficiency of our microscopes a bit more since this recording. And we've now done 24-hour recordings where we get only a few percent photo bleaching. And so we're kind of right at this edge of being able to push these recordings to just 24/7 uninterrupted recordings without bleaching the fluorophores, which is pretty exciting. As I mentioned, we've done a lot of work on minimizing the cabling because that's really important to get natural behavior out of the animal. But for some cases, you might want to completely remove any tethering that comes from the animal to off-board electronics. So we've developed these, what we call wire-free miniscopes. They have a battery that's oftentimes mounted to the side of the miniscope, and they log data to an onboard SD card. All of the electronics have been redesigned to make them as power efficient as possible. So the major weight of the system is the size of the battery. And so if we can make the system more power efficient, we can extend recording times or make the system lighter. So this allows us to do things like record in very large environments where the cabling, if you had a coax cable or any larger cable running off the animal, this environment wouldn't really be feasible. So this is about a 7.5 meter long track. There's a behavioral camera on each end of the track with a mouse running back and forth wearing a miniature microscope. Again, this is decamp imaging, but we pseudo-colored the cells and show some example traces. You can now also imagine using a system like this for imaging 2 animals simultaneously because cables won't get tangled up. And for some social experiments, I don't think I have the data in the slides, but we found that just removing the cabling, even though this adds a little bit more weight to the microscope, removing the cabling can enable some social experiments that you couldn't achieve with the cabling, potentially because the cable is distracting or provides kind of a looming cue to the animals that kind of inhibits certain behaviors. This is the same data. We can extract place cells and look at spatial coding as the animal explores these large environments, but for the sake of time, I'll skip over. So this is the version 3 wire-free miniscope. So it's like a previous generation wire-free system that's openly available, and a few companies sell kind of like modification kits to convert a wired miniscope to this. And we're just finishing up our kind of newer generation, which should be shared very soon, which is built on the version 4 platform. So a larger field of view, electro-tunable lens, should be a bit lighter or extend recording, and can be wirelessly triggered. So it has this little IR sensor, and with a remote control or a little device, you can mount this on a heavy animal and then trigger it to record and stop recording. We also are finishing up some work to integrate electrophysiology into Miniscopes. So we really like the coax cable. If you haven't said it enough, we really like having a thin cable coming off the animal. So whatever we want to add new features, like add a second LED for dual imaging, or add potentially some electrophysiology recording alongside the Miniscope, we really want to do this without adding any additional cabling. So the actual coax cable and the way it transmits data and power is quite flexible. So for all of our cases, we really designed the system to not add cabling and just pack additional data into the already existing cable. So this electrophysiology Miniscope just integrates an additional kind of expansion board built around a 32-channel Intan eFiz chip, and then has a little microcontroller that sort of synchronizes and packs the data in a way that can be packed alongside the imaging data and sent out over a single coax cable. And these are some of the first recordings. This was done by Sung in Pimon Galshani's lab, imaging CA1 with a Miniscope with a silicon probe designed by Sakiris Masmanidis' group at UCLA in the brain. And so here you can see some calcium imaging along with some channels of the silicon probe with some downward going extracellular spikes. We've also done this with collaboration with Tad Blair's group in rats. So they have a ton of experience with doing tetrode recordings, and now, which I'll show in a second, a lot of experience doing calcium imaging in rats. But so here you can see this was a few years ago. This was sort of the quality, not the best, but pretty close to kind of the average quality of calcium imaging in hippocampus that they could get. It's on the order of like 50 to 100 cells in a freely behaving rat, which just keep that in mind for when I show what we can do now. And then these are some of the tetrode channels that are implanted in the other hippocampus, and you can see nice LFP as the animal moves around and theta oscillation. So calcium imaging is great for kind of getting slower temporal imaging of neural dynamics, but you can't get oscillations in LFP. So even just not using all 32 channels, if you just have a couple electrodes that give you electrical oscillations, it can be really helpful for analysis. And we have we're finishing the newer version of this that will hopefully release soon, around the version 4 Miniscope that incorporates 32 channels of electrophysiology. The challenge of a system like this, especially for mice, is really not the electronics. It's easy enough to design these things, but it's the mechanical layout of how you fit a miniature microscope and electrode device on the surface of a mouse and be able to connect the two day after day reliably. So while we've had systems that are capable of doing this sort of recording for a few years, we really have gone through a couple iterations and hopefully have finalized the design now that really makes actually using a tool like this feasible. So hopefully that'll be released soon. And so let's see. So then some work that's just finishing up now by a postdoc in my group, Changlang Guo, was to build a larger miniature microscope for animals that are larger than mice. So for rats and non-human primates. So we call this the LFOV Miniscope. And this is now, large, too big to be worn on a mouse, but can comfortably be worn on a rat on the order of 14 grams and gives us a much, much larger field of view, still with single cell resolution across the entire field of view. So this microscope is really like a miniature microscope that we developed, but because we have the space and weight to work with, it has substantially better optics. So we have this kind of objective module that actually you can screw on and off just like a tabletop microscope and swap out the objective to change your magnification for your working distance. And then the main body of the microscope has a tunable lens, just like the smaller miniature microscope, so you can adjust focus, and then mounts an image sensor and some electronics, but just has a bit more optimized optics because we have the space for it. So this gives us a field of view that's much larger than previous generation Miniscopes. This is just an image through a cranial window in a head fixed mouse. So this is close to 3.5 by 2.5 millimeter field of view. In comparison, this blue square is the field of view of our kind of first generation that we released Miniscopes. This is the field of view of the largest commercially available miniature microscope. Orange is kind of the standard field of view of our new generation version 4 Miniscopes, and this gets you quite a bit larger. So you can do things just like record a lot of cells. This is, again, in a head fixed mouse, but with a microscope that maybe costs $1,000. This mouse is on a little circular treadmill, and we're looking at 1,900 active neurons that could be detected in this short recording, and looking at how these neurons are modulated with the movement on the treadmill of the animal. So we get quite nice optical performance, single cell resolution across the entire field of view, which is really great. But what we really want to use this for is in freely behaving animals. So in collaboration with Tad Blair's lab, we've done a bunch of imaging in hippocampal CA1 layer of rats wearing these miniature microscopes. And so this is just the raw imaging. If you remember a few slides back, I showed sort of what a few years ago where this group was at and miniature microscopes were at in the EPHIS video recording from CA1. And now this is kind of the kind of standard recording they can do. This is the 2 millimeter grid lens. So we're looking at a portion of the field of view of this large miniscope that just is filled up with a 2 millimeter diameter relay grid lens, and you can see cells flashing here. And now you can do things like have the animal explore a rectangular tract or a large environment while recording neural activity. Here, we're just plotting some of the cells that are classified as place cells that are highly spatially modulated that fire along this one arm on the right side. And you can see the sequence of cells light up as the mouse runs back and forth. We track the position of the animal with behavioral camera tracking the LED or the body of the animal using a machine learning technique. And then we can also get the head orientation with the head orientation chip on the animal's head. So that little yellow rat head is really combining all the data that we have while now imaging maybe 1,000, 1,500 cells in a freely behaving rat. Because rats and large animals can carry more than 13 grams, we also designed this little expansion board, which we call the WireFree Extension data acquisition system, that just can convert any originally wired Miniscope into a wire-free Miniscope. So it's just a small board that mounts on the side of a microscope, or you put it in a backpack. Again, it's battery powered and logs data onto a micro SD card. And this now just converts this large microscope now into a wire-free microscope. So, and because again, like rats can carry a decent amount of weight, we can add a large enough battery to record potentially for hours with the system. This just an example imaging from the WireFree system, example image as well as the cells extracted. Here we are looking at place cell activity in an open field. These are just 4 example cells, and they're kind of spatially modulated firing maps on the bottom. Also, this postdoc, just because it seemed possible, decided to try to make a smaller version of this large field of view miniature microscope. We don't really have a great name for this yet, but this is a large field of view microscope that's small enough to be worn on a mouse, so about 3 grams. That gives us about a 3 millimeter diameter field of view. And so this is the first freely behaving recording we did, so it's not super great, but this is just a cranial window with the mouse wearing this smaller large field of view miniature microscope, and you can see a bunch of tiny little cells flashing here. You can also do things like do dual grid lens implants. The surgery is kind of tricky because you have to get the surfaces of these lenses pretty parallel, coplanar with each other. But now with a single miniature microscope, you can image 2 grid lenses. These are both implanted over medial prefrontal cortex in both hemispheres. And then you can record from both. So I just zoomed in on these 2 lenses, and you can now see neural activity bilaterally with the system. That's pretty good. I'll finish for the last few minutes on what we're working on now. So one of the things we want to do is not just have a nice imaging of neural activity. We want to be able to actuate that activity and also improve the quality of single photon imaging in freely behaving animals. So we've started this project we call the Stemscope, which effectively takes a regular miniature microscope. We looked at the optical path of a regular miniature microscope. We have this LED on the left that sort of gets defocused through the optics and illuminates the tissue under the brain, and then cells fluorescence lights travels back up through the optics and forms an image on an image sensor. We're effectively removing, and we're going to end up redesigning the entire system, but what you're effectively doing is removing the single LED and putting in effectively what's a projection system. So a spatial light modulator that gets illuminated by LEDs, either a liquid crystal on silicon display or a DMB display. You can think of this really as just a tiny projector on the miniature microscope. This projector now can generate spatial illumination patterns that get updated at hundreds of frames per second. And those patterns get focused coplanar with the plane that the Miniscope is actually imaging. So we can project patterns at reasonably high frame rates into the brain and then image that activity that we're projecting in. So here we're just projecting this little cross onto, in this case, it's just a little GFP slice of tissue, and we only illuminate the places that we want. So I'll skip over all this, but it has a bunch of potential here. We can, in an automated way, identify cells and select which cells we want to illuminate. And you can do this with 2 colors at once. So you can imagine imaging a population of neurons while actively actuating specific neurons are close to single cell resolution in like a closed loop manner while you're imaging neural activity and behavior with optogenetics. The system, again, I'll sort of skip over it, but we can do some slightly fancier things with the structure of the excitation light we use to excite GCamp to drastically improve the quality of single photon excitation imaging of neural activity. So we can actually push past a lot of the limitations we have initially hit with just wide field illumination using some better approaches. This is just a recording in a head fixed animal showing comparison of wide field illumination versus just a simple mask. So we're just masking that pattern in the projector, so we're only shining light to where we have detected cells. And we get a significant increase in kind of signal to noise and size of neural dynamics. Okay, yeah, so finally, we build a lot of these tools that can image neural activity or record or modulate neural activity. But all these tools are really designed for freely behaving animals in mind. So this kind of a minimum usually involves some type of behavioral camera that gets synchronized with the neural activity. And so when we can, we really try to just make doing these experiments easier. So we built this thing we call a Minicam, which is just an open source behavioral camera that interfaces the same way as a Miniscope does, sort of just like a decent, easy to use, flexible camera that works reasonably well. We've also designed, and we keep developing, our open source acquisition software. So this can stream multiple video streams of Miniscopes and behavioral cameras or webcams simultaneously. We can do things like select little ROIs in the live stream of the Miniscope recording and plot the dynamics. It does real-time delta F over F conversion to kind of visually help see cells. We've also recently incorporated deep lab cut live into the software. So this is just would be a live video stream of my son walking in our kitchen. And in real time, we can do pose estimation through deep lab cut live while we're streaming other behavioral cameras and Miniscopes. And then Denise Kai's group, Phil, a grad student in her group, has developed an open source analysis pipeline called Minion that works with Miniscope data as well as other miniature microscope data. And while there's a bunch of these open source pipelines to kind of process your data and turn it into the neural activity of individual cells where you can start doing your own scientific questions, that pipeline is quite a large hurdle for labs to get through when they start with miniature microscopes. So the goal of this Minion pipeline was to kind of build in these really robust approaches to processing this data, but do it in a way that users that have never done this type of analysis or maybe aren't super comfortable in programming in Python can really understand what's going on under the hood. And there's maybe dozens of parameters throughout this pipeline in Minion or any of these other pipelines that really need to be dialed in for your recording and processing to work correctly. This sort of teaches the users what those parameters are doing, how it's affecting their data, so that you can confidently go through the pipeline and extract and process your data. So you can get that on the GitHub link on there. I think I'll stop there. We're moving towards trying to get as much data out of the animal as robustly as possible in as freely behaving natural environments as possible. So our group now is pushing towards trying to build fully wireless miniature microscopes. So wirelessly powered and wirelessly transmit data that can be chronically recorded onto a mouse in a colony of mice. And because we can now sort of image long-term without photo bleaching, to record uninterrupted for months as a colony of animals live in a natural environment. And so we're working on building these platforms now with the goal of really being able to do months-long neural and behavioral recording in ethologically relevant environments, and then making all that data and process data fully accessible to the neuroscience community. So I'll end there, so maybe we have some time for questions. I'll just thank my group, Changlang Guo, to develop all the large field of view miniature microscopes. I helped with a lot of the rat imaging. Federico does sort of a bunch of stuff across all these platforms. Maybe I didn't have the slide for the two-color scope, but he's developing the two-color scope. Raymond worked on a bunch of the WireFree stuff. And I mentioned Denise Kai and Tristan Schumann really developed the miniature microscopes with me when we were all postdocs at UCLA, and they're continuing at Mount Sinai doing their own development, like they have a two-color scope and developing the analysis pipeline and doing cool experiments. And then Payman Galshani, Alcino Silva, and Balji Kok are the three labs that this whole project started in when we were postdocs. And so this really got off the ground because of their vision of realizing that you don't have to pay $150,000 for a system like this, and then being able to build something that's not just used by us, but build it in a way that really any group can feel confident and afford using, I think has really been a really important vision for this type of work, and really came from them. So yeah, I'll stop there. We're recruiting. If anyone's interested in building these tools and using them, both of those things, we are recruiting for multiple grad student and postdoc positions. So please feel free to reach out to me. Again, with that, thank you very much. I'm happy to take any additional questions.
Thank you so much, Daniel, for that broad and really thorough talk about all the Miniscope universe. I think it will give everyone lots of technical details and with all the documentation you have and everything.
Great.
So actually, we have we are being showered with questions.
Okay.
So we might run a little long if
Okay.
you can handle that, yeah.
Sure.
And I'll try to organize them so we can answer all of them. So in terms of the long recordings and battery life, some people were asking whether you were working on streamlining data storage. That was from Amol. And whether Yao was wondering whether the batteries could be worn on a vest for animals to wear the battery of
Sure.
the body.
So mice don't like to wear a backpack or a vest. So for doing this kind of wire-free battery-powered recordings in mice, we really think pretty much the only option is to mount it on board the microscope. You know, there's other like low-power EPHIS devices that might subdermally implant a battery. The issue here is like these imaging systems, the bandwidth and power consumption is just larger than what kind of these subdermally implanted battery systems usually can power. So that's generally not an option because the battery does warm up a bit. And so I think for mice, really have to mount it on board the microscope. For larger animals, like rats are happy to wear a vest or a backpack. The whole system is still light enough to just put on the head of the animal, but it just kind of this wire-free expansion board just connects over a coax cable. So you could easily put a battery and that recording system on their back and, you know, record for, I mean, I would imagine in that case, you would have enough power and the micro SD cards are huge. So you could record for potentially, you know, a day if you didn't get photo bleaching. In terms of like optimizing data storage, so in terms of the wire-free setup, SD cards are effectively, in our case, unlimited in size. Like we're not going to fill up 128 gigabyte SD card, and the write speeds we can achieve are reasonable for the data we record. So we don't have to really, for the wire-free setup, do any data compression. Additionally, kind of doing any of that takes computational power, which takes energy and takes electrical power. But moving forward to like fully wireless systems, wireless bandwidth of transmitting data is really, you know, scales up, bandwidth scales the amount of power consumption used. So we are thinking about ways of doing clever data compression or some initial processing on board the device to decrease the bandwidth. If you just wanted to have smaller data sets, like our open source software does do lossless compression, which usually drops the file size by a half to 2/3. You can also do lossy compression, which usually doesn't work too great with neural imaging, but there maybe are some specific types of recordings where you can, you know, have a factor of 20 compression. But it also to say like data storage is cheap. So like even if you store completely raw data, it's unless you're doing, you know, dozens of animals with long recordings all the time, it's not a huge problem, I think, to get over in terms of just having a large data storage. But long term, yeah, there should be better ways of just recording, you know, terabytes of data.
So for some of the points you mentioned, photo bleaching and also the wireless aspect, Amon was asking whether there were any other calcium indicators that should be used or designed for long-time imaging, and also whether the wireless aspect of it was decreasing the stability of the miniscope on the animal's head.
So in general, like G-camps are, I mean, they improve every year. And like G-camp 8s are like ridiculously bright. So they are getting faster and more efficient and brighter. So in terms of if you wanted to go for the longest recording with least photo bleaching, the newest generation of G-camps is generally the way to go. And then the slower the version of the G-camp is, so going to like G-camp slow instead of G-camp fast, also has a pretty big impact in the amount of fluorescence you get for the input excitation. But I like the recordings we've done, which are these 24-hour long recordings, you know, we try to really make sure everything's optimized in the system to use as low power as possible. And we actually have our newest recordings, we actually hit the bottom of the LED drivers we use. So we actually kind of hit the default LED leakage current, and we can still actually slightly over bright for what we want. But that's solvable. but going to, and also the 24-hour recordings we've done are just using GCamp 6 fast. So like just going to 8 slow or 8 something will give you a, you know, a fourfold improvement of recording length. But yeah, there's also like we've imaged RCamp before. It's, you know, everything's, all calcium indicators are dimmer than GCamp, but RCamp you can generally nicely see as well. In terms of like wire-free stability and like a wireless Miniscope, you would so like the mounting of the Miniscope onto the skull of the animal for our newer microscopes are all a rigid connection of aluminum on aluminum. So the bottom of the Miniscope has an aluminum sleeve and the base plate's aluminum. And these newer generation Miniscopes have a set screw that locks the Miniscope into the base plate. And that set screw is a conical point set screw. When you tighten it, the conical point locks into an indent into the bottom of the Miniscope and basically rigidly registers the Miniscope in X, Y, and Z into the base plate. And really, you don't have any motion of the Miniscope relative to the skull of the animal for a wired or wire-free setup. The motion you see is really the brain moving relative to the kind of the rigid skull miniature microscope. So I don't think stability of the system is sort of an issue. If anything, it's better because you don't have a cable running off the animal's head for these wire-free setups.
Thanks. So I was wondering about the weight. I think the lowest, you mentioned all these larger systems, but for the lowest ones, you were down to 3.3 grams. For songbirds, we work around like a max 10% body weight, 2.5 grams. What can be done there to reduce the weight, or have you tried with songbirds?
Yeah, so the older generation Miniscopes with 3.3 grams, our newer version 4 Miniscope is 2.6 grams. So that's the kind of large field of view electrotunable lens setup. Yeah, so especially for songbirds or juvenile mice, you would want to go potentially even lighter. We have some designs, I forget what we call it, but we have a design that's like a 0.8 millimeter, 0.8 gram miniature microscope with some of the electronics offloaded from that to like a little backpack for a songbird that we haven't finished building. That seemed to work. But there's also like, so like the Miniscope is this open source miniature microscope from Tico Hoogland's group. That's can be, 2 of them can be mounted on a mouse, and they're like maybe 1.8 grams, 1.6 grams. So that would work potentially well for songbirds. The big difference in our setup and theirs is they kind of reduce that weight by not having the electrowettable lens. So there's a manual focus instead. Michael Fee's group just put up a paper a few weeks ago on bioRxiv in what they call the feather scope, which is like a 1 gram scope, sort of for songbirds, as well as they built a larger field of view, lightweight miniature microscope as well. But it's possible. We sort of are generally interested in building things that have that we are our lab ourselves, or we have a close collaborator that needs sort of like has some hurdle in the way of doing their research, and then we like to work with them to address that hurdle and design systems around that. And we just haven't had a close collaborator that needed a system like this for small animals or for songbirds. Otherwise, I think it's very doable even for our platform as well. But again, there are systems, open source systems even that exist that are a really good fit that are under 2 grams already.
Thanks for that comment. Actually, in terms of these collaborations and different microscopes systems, Andre on YouTube was wondering why these different groups are not working together, or if they are, how they're working together. And also for the commercial options that you mentioned, what the companies like give back to the project, or how does like the Miniscope project feel about the commercial aspect of that? So these are tough questions.
No, I think that's a great question. So I would say in terms of academic groups developing miniature microscopes, which are pretty much all the academic groups are making open source microscopes, we all like do somewhat work together. Like I think we're super friendly with each other. Some of the systems are built off some of the electronics we've designed. And I think we sort of all know each other and are very happy to work with each other. We usually end up kind of working on, we know what roughly everyone's doing, and we try to sort of are addressing our own sort of interests and challenges in the field. So there's oftentimes, overlap, but not complete overlap in what we're working on. But I think the open source field in general is extremely friendly, and we definitely work together. In terms of commercial companies, there's a few, right? So like the main one people probably know about is Inscopics, which was started by Mark Schnitzer's lab and some of the people in it that initially really developed miniature microscopes from the 2011 Nature Methods paper. There's Doric lenses, Doric optics, that also make miniature microscope. There's like Neuroscience that makes a fiber bundle-based miniature microscope. We generally have really good relationships, like Doric and Neuroscience. We are always, they're both very open with us and we're open with them, and we help each other out. Inscopics has been not necessarily that case, but I think there's absolutely a place for commercial access to these tools, and open source tool development doesn't fully fill the need of what commercial development can provide labs. We do, we're really trying to push forward in kind of making open source not just a term you say and put design on GitHub, but we really think for some of these tools like miniature microscopes that they should be able to fully kind of fill a lot of the need that potentially in the past commercial companies filled. So again, working with OpenEtha's production site, who are just fully committed to like this open source vision and really thinking that for some cases, open source is absolutely the way to go. They are really pushing this idea forward, and they provide kits or fully assembled miniscopes, not just our stuff. They provide other open source tools as well, but also like warranty and support. And so I think if the lab has the money and not the potentially like resources or manpower to debug a system, maybe a commercial approach is fine. But I also like the idea that if you built a miniature microscope, like it's yours, you own the thing, you understand what went into it, so you can design your experiments better because you understand the capabilities of the system. Since you built it, if something breaks, like nothing on the system breaks really that takes more than a minute to fix, but you don't have to be worried about working with the tool you built. And also, once you understand what goes into the system and when you built it, it maybe leads groups to not be so worried about modifying the system to fit the exact experiment they want. And so our part of our hope going forward is that labs start to really understand the tools they're using, and now it helps them think about what's the best experiment they can do, and then take the tools that they understand and modify it. Like swap out the filters, and now you can image red instead of green, or look at the other dozen open source miniature microscopes that are built on similar frameworks to understand how you can incorporate some of that into your work. I think that can oftentimes lead to better science than being limited to specific tools that are sitting on the shelves in your lab. I'll finally also say that for some of these tools, like miniature microscopes, they don't have to be expensive. And because our miniature microscopes are 50 to 100 times cheaper than closest commercial option, it does that's just the cost of making them. And luckily, NIH and NSF have seen the value in open source tool development and invested in this project. And that really makes it so that it's not just the top 5% of labs that have $100,000 to buy a tiny microscope for, but it's all the other labs that can afford spending $1,000 or $2,000 to get these tools into their labs. And then you learn how to use them. And that information of how you build these tools and how you use them and modify them doesn't sit at a company. That information gets learned in labs that I think kind of translate to much better experiments down the line. So I think for all those aspects, I really think open source development is useful. But again, we're not in any competition against any commercial company. I think there's a need for that as well.
Definitely. I think he was mentioning the commercial sense of selling miniscopes or lab makers and OpenEffis that you mentioned, but you touched upon that, saying that they develop stuff and feed back into the project.
Yeah. So all our approach open. So we don't. Our group and the Miniscope project itself doesn't directly sell anything. We kind of develop things on an academic way, but we want to make sure that these tools are actually disseminated and can be used. So with LabMaker and OEPS, we work with them to make sure, they have to be interested in actually like bulk ordering and supporting these tools and assembling them or putting together kits. But they handle all of that. Our only role is sort of like, if we're making new modifications, we make sure they sort of know. If they get feedback of like issues in the design, they feed that back to us. And then like, we want to really make sure these are one, reliable for people that get these systems from anyone. We hope that they're reliable. And we want to make sure they're affordable. So if a company was to try to sell these for more than we think they should be, we might actively try to make sure that there are avenues that are priced at a place that we think is reasonable. But the lab maker and OEPS are very much open to that, and which is really great.
So maybe just to touch on the last subject, these 5 more minutes, people were wondering about data analysis pipelines, whether you were working with some data analysis software that was, or developing, that was Andre. Then Amol was asking whether the Miniscope data can be exported and analyzed in commercially available software. And Mikkel was wondering about how Miniscope and Minicam interact with Bonsai, but I think someone in the chat already pointed them to the wiki link.
Cool. Yeah, so I'll just say, like, with the Nice's group and Phil, we have helped develop Minion. So that's this open source analysis pipeline for miniature microscope data. The recordings from any miniature microscope ends up just being some type of video file, right? So I think like maybe in Scopic's Mosaic software, I actually don't know, I've never used it, but I think it maybe works with TIFF So you can always attempt to, I would imagine, convert miniature microscope. We record usually in AVI files, convert that to TIFF But I think for commercial groups using commercial systems, as well as groups using open source systems, I think like the vast majority, I would guess, use these open source pipelines. So Minion's one that's relatively new. There's Kaiman, which is probably like the most well-known, supported and maintained from the Simons Foundation. And then for a MATLAB-based version, the package called MiniPipe, as well as a few other ones. There's some analysis pipelines from McGill University. And so I think those all exist. They all are doing under the hood something pretty similar. But the real trick is like, I don't think there's any solution that is like a black box that you can plug your data in and it outputs reliable, neural activity out. Even if the output looks good, looks like individual neurons and looks like calcium dynamics, many of these pipelines kind of force the output to look like that. But it's still under the hood or directly on the surface. There's many parameters that very much affect your data. So using any of these approaches, Miniscopes or other miniature microscopes, I think you really have to have a good understanding of how you go from raw data to like neural activity traces. And hopefully Minion is one of these approaches that really help that with that. There's a Jupyter notebook, and every chunk of the process, you see the code with a big description of what the code's doing. And you can run the code using multiple parameters, and then it visually shows you the impact of those different parameters on your data and explains what you were looking for to optimize the data. But I think for the most part, they're all open source. They all are doing something similar. You can always convert your data into whatever format these pipelines take. But I think it is like a somewhat of an open question and things that people are working on of how, like, in the end, building Miniscopes, labs are sort of worried about like taking an assembly kit and like putting lenses in it and working with a circuit board. That ends up being pretty easy. Labs then are really concerned with like how do you actually do the surgeries, but mostly most labs have worked with animals, done maybe EPHIS surgeries or other optical implant surgeries. That ends up working fine. You then have to get the animal to perform behaviors and stream data, which also labs seem to end up getting over that without too much of a problem. But analysis and understanding analysis is really like remaining hurdle for a lot of these labs, especially because a lot of them, maybe this is the first time they're working with these large neural imaging data sets. So I think there's still work to be done to really push how labs can use and understand and validate these analysis pipelines moving forward. And we're working on that, but I think a bunch of other groups are working on that as well.
So thank you so much for all of your answers. We really kept you a long time. You're welcome to stay, everyone. If we're going to shut the live stream now. And next week, the week after, we're going to have Dr. David Kennedy talking about repronym on this series, and you're welcome to join again then. Thank you so much.
Okay, thank you very much.
That's it, we're off live.